you have here a live demo of a T cell, T helper cell, and can you share that with us and just tell us a little bit about the process and what you're doing? It's Alex.
Yeah, I'm just. Please let me know when you see my screen.
We see it.
We can.
Perfect. Just 1 second. Okay, so just to give you, I don't know if you can still see my face, but
yes,
we can.
Okay, perfect. So you see, like I'm wearing this a headset. So this is an Oculus Rift S, and you can see I'm holding these two controllers. And if you look at the screen, you can probably see two kind of blue-ish controllers and the two kind of like spheres. So these are the direct representation of what of my hands, and what you see on the screen is exactly what I see in the headset. So this is just to give you a bit of context. What we have here is the helper T cell that Steve's lab imaged. So we can kind of like manipulate the data, move it around. We can also go inside it and see what's happening, the thickness of the membrane. We can kind of like start doing things like kind of really cutting it, slicing it, seeing what's happening in different areas. And we have like a range of different tools in here to be able to analyze this data. And this is just before I stop talking, but this is the axon and the spectrum rings that Steve was just talking about. So this is data from the Le Terrier lab. I'll just make it slightly bigger. Cool.
Can I just ask, did you guys, did you have to fix these cells? How did you prepare them before the visualization even happened?
Yeah, so these cells are predominantly nearly all of these are fixed. So these are plated neurons, I believe, they're fixed. To actually to break the diffraction limit to be able to do super resolution imaging, the trick that one has to play off to avoid violating the uncertainty principle is to trade off spatial information for temporal information. So what you're actually doing here is it takes a while to acquire these images, typically on the order of minutes to hours. So what we're not doing here is seeing a kind of a live reproduction of living cells. That's certainly something we'd love to do, and we are working towards. Certainly the software here, vLUME, will be capable of doing it once we get once we go back in the lab and work really hard to be able to do that. And we have certainly some ideas about how to do that. So that's certainly a place we want to go into the future. But these are kind of static three-dimensional images of, in this case, a neuron, but also the T cell membrane that Alex was just showing.
It's fantastic. It's really beautiful.
Yeah, I mean, maybe I could just give a bit of a scientist perspective here. So, we generate these data, and actually the output of a super-resolution microscope is not a picture, as you might think of a standard microscope. What it actually outputs is a text file, a list of the spatial position of individual fluorophores. And then what the software is doing here is rendering the positions of those fluorophores for you to be able to, in your brain, interpret that as a three-dimensional image. Now, you might just say, okay, well, you could just plot that. It's just a three-dimensional plot is what it is. And you're absolutely right, you could do. But what we found, and actually what's been so wonderful collaborating with Alex on this project, is that what this allows researchers to do is really explore their data in an extremely intuitive way. So first off, that biology occurs in 3 dimensions, even though that the vast majority of microscopy images is 2 dimensions. So your confocal microscope might take a 3-dimensional picture, but probably, which is itself a series of 1-dimensional pictures that it maps together. This is actually taking a 3D picture and rendering it. It allows us to explore that data and really try and see it from different perspectives. And that kind of immersive nature of virtual reality and the ability to see it from different angles. First off, there's a variety of things. One of them, it allows you to examine the data to make sure it's of high quality. It allows you to segment specific areas of that data to be able to make sure that and perform quantitative analysis on various areas of it to make sure that one area is similar quality to a different area. And also, what we've really found is, more importantly, it's really allowed us just to communicate our discoveries back to our collaborators and to other people in quite a timely manner and quite an efficient manner. And actually, these three-dimensional data sets that we have, it's very difficult to take a 2D picture and really get the intricacies of what we're actually viewing. And VR, I think, does provide a unique opportunity. What we did find was that literally everybody, all of my graduate students who've been taking these kind of data, when they were first examine their own data that they're very familiar with in virtual reality, nearly all of them were blown away and saw something new that they hadn't seen before, even though they were really familiar with looking at that particular data set. So it's really, you think, okay, it's just, you're just visualizing the data in an easy way, but that itself has scientific merit because it allows us to reject hypotheses quickly. It allows us to create hypotheses, and it allows them to test them and share them with our collaborators.
Yeah, the shareable, functionality is really interesting. And the fact that even right now during this pandemic, people are very digitally connected. This offers another tool where you could, really in real time share something of interest and possibly, one scientist, looking, or two different scientists from different disciplines looking at one sample might have, glean different information from it. So it's really fantastic.
So if anyone's watching this video and would love to see that play with their own data in this way, that's perfectly possible. What one needs is the software. They need a VR headset and the computer associated with that, and the data off of any super-resolution, localization-based super-resolution microscope.
Fantastic. Thank you all so much. Alex, and Stephen, you have been, this is revolutionary. This is, and you've been so kind to share it with us, and we're hoping to promote it out to all the researchers we are in contact with, because I think that this is a really, I think you're at the beginning of something just huge, and it's a great tool. And I think it could really, provide a lot of new insight into T cells as beta cells, many cells. So thank you so much for sharing your time and showing us the demo today. I really appreciate it.
Thank you.
Thanks for having us.
Thanks a lot for your time. Thanks a lot for inviting us.